Okay, imagine you're on a walk and suddenly a mosquito comes by and bites you. Usually what happens is a small red itchy bump occurs, which normally subsides after a couple of days. But have you ever wondered what occurs within your body when this happens? As you get bit, your body's immune system kicks into action, fighting the pathogen. The human immune system is a complex network of organs, tissue, physical and chemical barriers, and lymphatic vessels that alert other cells and tissues when something is recognized as non-self. It consists of the innate immune system, the first and second line of defense, and the adaptive immune system, the third line of defense. Your first line of defense is non-specific, such as physical barriers, your skin, and chemical barriers, such as tears. For example, if dust or other small particles get into your eye, your body produces tears as a mechanism to flush out the particles. Physical barriers such as cilia prevent dirt, bacteria, or other invaders from entering your body, acting as a barrier. So what happens when the first line of defense is ruptured? Well, that's when your body's second line of defense is triggered. The second line of defense is also non-specific, such as immune cells like macrophages and dendritic cells. Macrophages, mostly found at the site of infection, are large phagocytic cells, whereas the purpose of dendritic cells is to process antigens and represent them on the surface of the T cells. The dendritic cells are the main connector between the innate and adaptive immune system. The innate immune system is genetically pre-programmed, denoting to the immune characteristics that the individual is born with, which does not require any learning or memory. Now that we have a basic understanding of the innate immune system, let's take a look at the adaptive immune response. The adaptive immune response is specific and moves into action when the second line of defense fails to annihilate the pathogen from the body. This system consists of the humoral response, which is ideal for pathogens within body fluids, and the cell-mediated response, which is specific to intracellular pathogens. When the second line of defense fails, it allows a pathogen the opportunity to access the bloodstream. The bloodstream can then carry the pathogen around the body, surrounding the spleen and lymph tissue. This is when the adaptive immune response is initiated. The adaptive immune system is classified as two main branches: the humoral response and the cell-mediated response. The cells in charge of assisting this immune response are called B and T lymphocytes. B lymphocytes attack antigens outside of the cell, whereas T lymphocytes attack antigens within the cell. B lymphocytes create antibodies which fight against pathogens that enter the body. They develop in the plasma cells and produce gamma globulins that identify antigens, binding themselves onto them. This process is referred to as a humoral immune response. T lymphocytes, on the other hand, convert cytotoxic T cells, which then search for infected cells, binding to them and destroying the cell. This is referred to as a cell-mediated response, as it takes place within the cell. The humoral response dispatches antibodies, monitoring the body's blood and lymph. As B cells or B lymphocytes grow, they are able to determine friend from enemy, developing immunocompetence as well as self-recognition. Antibodies are proteins within the blood that assist in battling against antigens. The unique shape of each antibody allows it to bind to specific antigens, where the macrophage then engulfs and breaks down the antigen using enzymes. The antibody binds to the antigen like a lock and key. Once an antigen is found by the antibody, it attaches to it, signaling for a phagocyte to engulf the antigen, activating the B cell. Once the B cell is activated, it produces multiple copies of itself, consisting of the same antibodies which are needed to fight the antigen, producing effector cells and memory cells. The effector cells are packed with large amounts of endoplasmic reticulum, producing antibodies at a quick rate. Whereas the memory cells record the antigen and its behaviors, preparing a secondary immune response, allowing the body to fight and annihilate the specific pathogen in the future. Through a process called neutralization, the antibodies physically obstruct the binding sites of the pathogen, preventing it from attaching it to the tissue. The structure of an antibody is specific to its antigen. The helper T cells are activated when they bind to protein-containing antigens, which then secretes molecules known as cytokines. These cytokines assist other cells by binding to receptors on their surface, regulating an immune response. These signal responses such as inflammation and swelling. The cell-mediated response is similar to the humoral response, but differs in which it eliminates intracellular pathogens. Specialized T lymphocytes target infected cells and destroy the entire cell. For example, the T lymphocyte may target cancer cells via receptors that allows the T cell to recognize and eliminate the tumor. Cell-mediated response specially targets and protects the body against protozoa, bacteria, virus, and tumor cells. T cells can identify fragments or parts of an antigen on the outside of a cell or within macrophages. These smaller fragments can be identified by protein molecules called major histocompatibility complex molecules, MHC. There are two types of self-identifying molecules which possess specific binding sites for antigens. MHC1 represents antigens that originate from the cytoplasm. They are expressed on all stemmatic cells and allow the T lymphocytes to detect any infected cells or macrophages. MHC2 are found on antigen-representing cells. If the cell is recognized as self, then MHC2 dispenses peptides that indicate the cell as own. The antigens engulfed by macrophages is then recognized as antigen representing. Therefore, the helper T cells recognize that it has a receptor, signaling the antigen that correlates with the antibodies. Cytokines produced by helper T cells initiate multiple of the similar helper T cells as well as macrophages. Then, a cytokine chemical called interleukin-2 initializes the multiplication of B cells, which are clones of the specific antibody needed to fight the antigen. As the immune system collectively succeeds in fighting the pathogen, suppressor T cells are in charge of discontinuing the production and activity of B cells and cytotoxic T cells. The adaptive immune system is specific as it targets and eliminates pathogens more accurately and thoroughly. With the help of memory B and T cells, the adaptive immune system is able to prepare secondary defenses for a specific pathogen. This allows the body a head start in fighting with the pathogen in the future.